Bismillahirrahmanirrahim. This is 9700 A-level biology. We're going to do the June 2020 question paper 4.1, and we're going to see how we're going to handle this. And some of the questions I'm going to do because I'm not going to do all the questions because some of them are easier and some of them are more difficult. So I'm just going to do the ones which are more difficult. Now let's go through question 1. In mammals, the hormone ACTH is released from the anterior pituitary gland, stimulates the cells of the adrenal glands to synthesize the hormone cortisol. So basically, when you're reading this question, you've got to understand is they're asking you basically, cortisol is not in the syllabus, but it is generally what is in your syllabus is adrenaline and glucagon. And they're going to ask you a question related to that. So this is an example of cell signaling. The main stages of cell signaling involving ACTH are similar to the main stages of cell signaling by adrenaline. Now, that's adrenaline is in your syllabus. Describe the main stages of cell signaling involving ACTH. So they want you to just talk about ACTH, but it is the same as adrenaline. So now you look at the marks and you think of 5 mark scheme points. So number one, you know a hormone has to bind to the receptor. I'm sure everybody knows that, but you're not going to write it. So then the G protein activated, the second point, and adenylyl cyclase activated, then cyclic AMP made from ATP, and cyclic AMP is the second messenger. So you got your 5 marks. If you wrote these 5 points, you got your 5 marks. But the others, maybe somebody didn't write this and then they wrote something else, is like cyclic AMP binds to kinase enzyme, then there's an enzyme cascade, and this amplifies the signal and results in the synthesis of cortisol. Why? Because this was given in the question. This was given in the question. It said synthesizes the hormone cortisol. So this was given. It says in hormone ACTH is released. ACTH stimulates the cells of the adrenal glands to synthesize hormone cortisol. So look at the last mark scheme point is results in the synthesis of cortisol. This was the last mark scheme point, and I'm sure anybody had written that would have got the mark for it. But you see, the main thing is in the exam is how you read the question and how you attempt. This is the most important thing when you're taking the paper 4. It is not how much you know, it is at that very moment in those 2 hours how well you attempt the question. That is the most important thing. Part of the question was if the concentration of cortisol in the blood increases above its set point, it will inhibit the production of ACTH by the anterior pituitary gland. Name the type of homeostatic mechanism. Naturally, when something goes above the set point and then there's a correction and that results, and that is of course called negative feedback. Now, of course, you would confuse this with homeostasis, but homeostasis is the maintenance of a constant internal environment. This is not that. This is, it said, if there's increase, and then of course this will not be produced. Just like your blood glucose level rises, insulin is produced. But if the blood glucose falls, well, insulin will stop being produced. So this is what you had to understand, is that the answer to this was name. Name means there was just one name to it. So you should have thought about it. Set point, increase, decrease in the set point, return to normal is negative feedback. And the C part of the question, the domestic dog, whatever, is a mammal. A dog may develop a condition called Cushing's disease, which is a result of too much cortisol being secreted by the adrenal glands. The most common cause of this is an increased secretion of ACTH because of a tumor in the anterior pituitary gland. Now, the direct question, explain how a tumor develops. Uncontrolled mitosis, abnormal mass of cells formed, cells are immortal, mutation of the tumor suppressor gene. So even if you don't remember this is actually something from the AS level. Tumor means an uncontrolled cell division and a mass of cells is being formed. So you had to remember that, and you would have got these 2 marks, 2 out of 2 for writing that. Now the D part of the question was, one symptom of Cushing's disease in a dog is that the dog will want to drink much more than normal. Drink much more than normal. Suggest one reason why a dog would want to drink much more than normal. I mean, immediately the thought which came to my mind was diabetes. Or maybe why? Because when do you feel thirsty? When you've had chicken kadhai and naan and coke, and you've had something to sweet, so your blood becomes more concentrated. This, of course, is a decrease in water potential in the blood. So that's why you feel thirsty when you've had such a heavy meal. Or eating food with a high salt content. If you have a lot of salty stuff, Well, you're going to feel thirsty. Why? Because suddenly your blood plasma is become very concentrated. So if you know bio, if you know your basic concepts, well, you can answer most of these questions. To question two, the patty pan, squash plant, whatever, produces edible fruits that vary in color. The color of the fruits is controlled by two genes, big A small a and big B small b, that occur on different chromosomes. So one is on one pair and the other is on the other pair. Now this is going to show independent assortment, because if they were on one chromosome, then it would be linked genes. So maybe A is on this one, and then small a is on this one, and big B is on this one, and small b is on this one. So this is what we have to say is controlled by two genes that occur on different chromosomes. So you've got to understand what does it mean when they're saying different chromosomes. So that means it's going to show independent assortment. Okay, then let's look at the rest of the question. It says allele A, big A. So then let's give it a color. Big A produces white fruit color. So what will be the white plants be? White plants will all be big A, or big A, small a. But then allele A, now the small a. Now when you look at what they're saying in this country, the question is in small a does not produce a color by itself. So small a are colorless, but allow the colors coded by the gene big B, small b to show its phenotype. So if there is big B or big B or big B, small b and small a, so then they're going to show this genotype. They're going to show the effect of the big B or the big B, small b. Then how more information that we have, then we have a slightly more information. It says if the plant is big B, here it says allele big B produces a yellow fruit color. So all those which are this or all those which are this are going to be yellow in color. Now this is all the information from the question. You need to figure this out. You need to be really analyzing this and you need to be figuring this out. So big B, but then it says, but what happens when there is, so if there is small a, then it does not produce a color by itself, but allows the color coded by this gene to show its effect. So big B produces yellow, and small b, what does small b result in? So small b results in a green color. Now that is what is the information given to us in the question. It says if it is small b, then it results in green fruit color. So this is how you first understand the question and then you proceed on and start doing the question. But this is the story you must do to yourself when you ever read the genetics question. Now as the question proceeds, in a dihybrid cross, big B plant was crossed with small a. So this is homozygous dominant for both characteristics, and this is homozygous recessive for both characteristics. All the resulting F1 plants produced white fruits. So okay, so all the resulting plants will be what? There will be big A, small a, and big B, small b. Why? Because what did we have? We have big A, and big B crossed with small a, and small b. I mean, sometimes, sort of, if you can't do this sort of a situation, what you can do is you can separate them. Like I always sort of, when I am mentally thinking, I say, okay, big A into small a. So let's look at this one first. So what are all the offsprings going to be? They're all going to be this. And then let's take this separately. Big B into small b. So all of them are going to be big B, small b. So please try to understand this and then it becomes very easy. So then they were all crossed with each other to obtain the F2 generation. Now complete figure 2.1 with the gametes produced by the F1 parents, the F2 genotypes and the F2 phenotypes. What information was given in the question? They gave you the genotype, so this is given to us. Now, I've always told you how to do this is there is a way of how you do this. And if you number this 1, 2, 3, 4, so then we do 1, 3. 1, 3 means big A and big B. So this is what we have here, big A, big B. And then we have 1, 1, 4. So big A, small b. And then you have 2, 3. And then you have 2, 4. I mean, this is the normal way that we do the gametes. So big A, big, so you get all this. We get big A, big B here, and then we get big A, small b, and then small a, big B. And then we, of course, get all these. But then look at how do we have to first get all the genotype right. That will get you 1 mark for getting all the genotype right. So if I say big A, big B is crossed with big A, big B, so big A, and big A, big B. And so we do this. And then what do we find? We find that we have 12 white. So there are 12 white. So there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. And then we have 3 yellow. So we have 3 yellow. This is yellow here, and yellow here. And then we've got 1 green. So, I mean, you have to really understand the genetics. And then, of course, you can do this question. It doesn't become very difficult. And so then we come to the ratio of the fruit color. So 12 white, 3 yellow, and 1 green. So please do this in your notebooks and then figure this out. Why am I saying that? Because if you remember, we did this in the beginning. When you have small a, if you have small a, then the dominant trait will, the B will show its effect. So in these 4, we have small a, this one, and this one, and this one. So in that, whatever is in the big B will show the yellow color and the small b will show the green color. So please understand this question, and then of course we proceed on to the rest of the question. To the part 2 of the question, test cross. Now test cross is that which is always done to find out the genotype of the mystery organism. Is it big B, or is it big B, small b? So you cross it with a known homozygous recessive. So the known homozygous recessive would be this. So it says test cross were carried out on 2 white fruited plants, P and Q, from the F2 generation, and each of these plants had its female flowers pollinated with pollen from a green fruited plant. Green would be this. Now plant P were white and were yellow. Q had some white and some green. Now you know green was only possible. Green is only possible when it has to have both the small a's. So then we needed to figure this out. So plant the genotype. Genotype is always written in letters. So P was big A, small a, and big B, and the Q was big A, small a, and small b, small b. So this is what you have to figure out. If you have to revise test cross, you need to revise test cross. Then it says the next part 3 of the question, plants P and Q show genetic variation with respect to fruit color alleles. Identify the process that occurred during meiosis in the F1 parents that produce this variation at state of meiosis at which it occurred. So process is independent assortment, stage of meiosis is metaphase 1. In part B of the question, it says in 1994, 2 new varieties of squash were grown in the USA. Both varieties showed resistance to 2 viral diseases of squash plants. The variety Tigress was developed by selective breeding, and the variety Freedom 2 was developed, or Freedom 11 was developed by adding genes for viral coat proteins to the squash genome. Identify with reasons the variety of squash that can be described as recombinant. Recombinant is the one which has been produced by a genetic technology. So Freedom 2, why? Because DNA from 2 different sources and was done by genetic engineering. Part 2 of the question was outline one social advantage and one social disadvantage of the recombinant squash variety compared to the non-resistant squash varieties. Number 1, naturally, we're going to improve the yield, so we're going to have more money and we're going to have more crop. And the disadvantage was that we don't know when we genetically modify them, that what these things that we've added, how it might cause allergies, how it might cause, because these are edible crops. So edible crops means that human beings eat them, and we don't know what harm it could do to them. Maybe after consuming it for 5, 10 years, people develop allergies, or maybe people develop kidney stones, or maybe people develop hypertension or diabetes or more cancer. So we have no idea, but this is all a lot of uncharted territories where we are just experimenting, and we have no clue what it could be doing damage to people who consume these products. Now coming to the third question, cats are members of the Felidae family. 2 genera of Felidae are this and this. The genus this consists of species of wild cats that are small and spotted. In 2013, biologists investigated the evolution of Leopardus trigenus in South America. Figure 3.1 shows the locations in South America of 2 populations of population A and population B. So they're two different places. Population A is in a certain part of the country and population B is in another part of South America. So population A lives in grassland and desert habitats. So they've given you a little more information now. So population A lives in grasslands and desert habitats, and population B lives in forest habitats. The cats in population A have a lighter coat color. So A have a lighter coat color and a different pattern of spots from the cats in population B. So the B and they have a slightly different coat color and different pattern of spots. Now, genetic analysis shows that population A is genetically distinct from population B. Population B has now been reclassified as a new species. Direct question, define the term species. So there is no sort of ambiguity. There is always a certain portion of the paper 4 which is a direct question. So this about maybe 25 to 30% of the paper. So they are direct questions and you should be getting your full marks on that. So define the term species, group of similar organisms, interbreed to produce fertile offsprings, occupy the same niche. Any two of these and you got your 2 out of 2. Then the part 2 of the question, explain how the two species, you may read the question or you may not read the names. These are difficult names, and I find when I read them, I just get more confused. So I just say, okay, LT, LG. So explain how the two species LT and LG have evolved from one original population in South America. So now, whenever there is the question on speciation, these are the points which you can write nearly in every paper. Geographical isolation. And if you know that name of that geographical isolation, it is called allopatric speciation. So if you just wrote allopatric speciation, which is a result of geographical isolation, well, you got your 2 marks. Different selection pressures in both the places, different weathers, climate. Selection pressures means weather conditions, climate, food, plants. So different mutations, some mutations make individuals better adapted. Those individuals survive, pass on the advantageous alleles. Now, this is a very typical biological term, pass on the advantageous alleles. It's not genes, it's alleles. We all have the gene for blood group, but we have different alleles. I'm blood group O, so I have IO, IO. Somebody else is blood group B, so it's IB, IB. Somebody is AB, so it's different alleles. So this happens over many generations, and these are the sort of points which you can write in every question which is on speciation. And these are the ones which you need to remember and memorize so that you can write them whenever there is a direct question. And this was for 5 marks, but the mark schemes are of course very larger numbers available for you to get your 5 out of 5 marks. And coming on to the B part of the question, it says illegal trade threatens the survival of endangered species. Forensic tests can identify animal species from the DNA of their body parts, for example, bones. Cat species differ in the number of repeats of bases C and A. So these are called the CA repeats with one particular region of DNA known as PLE46. Table 3.1 shows the number of repeats in PLE46 for 3 endangered cat species. So Asiatic lion 22, Bengal tiger 7 to 8, and leopard 14 to 15. Gel electrophoresis is used to estimate the length of PLE46 in a sample of DNA to check whether the sample comes from one of the endangered cat species in table 3.1. So outline, again direct question, outline how gel electrophoresis is carried out to confirm. Outline means just the basic outline, not the details. Whether the sample comes from one of these endangered species. So DNA sample placed in wells, electric current passed, DNA moves to the positive electrodes. Why? Because DNA is negatively charged. And the shorter fragments move further in the unit time, and you compare the band positions to identify the cat species because it will give you a different pattern of banding. So this is how you will identify the different species. The last part of the question was custom officials are responsible for checking whether animal body parts come from species on the CITES list of endangered species. Official can now use a small DNA barcoding kit linked to a computer database to identify a species. Computer database, bioinformatics, this is all something which is part of the syllabus. Suggest two advantages of using this method. It's a very quicker test and it can be carried around, so it's portable, quicker, cheaper for the low-income countries, and many sequences can be stored, which has a database. So bioinformatics comes in here, and it is for 2 marks. You have to give me some sort of information showing the advantages of what they had told you in the question. So small DNA barcoding kit linked to a computer database. So that was all very simple. So that you would do the test and then you would compare the barcode with, and you'd go on the website and compare it. So it was a very quick test, cheap, portable, and many could be stored. Come to the question 5, which is probably the last question we do in this video. Yeast cells are unicellular eukaryotes. Yeast cells respond to changes in the concentrations of the sugars, galactose, and glucose by switching on genes using transcription factors. So let's read the question again. And yeast cells respond to two substrates, galactose and glucose, by switching on genes using transcription factors. Figure 5.1 shows the events that occur when the sugar galactose is present and the glucose is absent in the external environment. MSN2 transcription factor proteins bind to promoters of genes A and B. Gene A switched on, enzyme A synthesized, galactose converted to glucose. In the other situation, we have gene B switched on and protein B synthesized. So please read the question, understand it. MSN2 is a transcription factor protein which bind to promoters of genes A and B. So when it binds to promoter, naturally mRNA will be made and those proteins will be, so transcription translation will take place. Now, the first part of the question says, explain why enzyme A is described as an inducible enzyme. So basically, if you know the definition of an inducible enzyme, you can write this. Inducible enzymes are those which are not made all the time. And these genes are switched on when needed, triggered by the absence of glucose. Concentration increases when galactose is present. So, inducible enzymes are those which are not made all the time and they will only be switched on when needed. And they will, of course, here because triggered by the absence of glucose, no glucose. So they will start to be switched on, and then concentration increases when galactose is present. Then we come to the part 2 of the question. Protein B has a similar role to the protein coded for the gene Y of the lac operon. Now that, of course, was the permease one. So suggest a possible role for protein B that would allow yeast cells to make use of galactose. It's a channel protein for galactose so that it can enter and be used by this. Once it enters through this channel protein, if the channel protein must be in the cell membrane of the yeast cell. And that is going to facilitate the movement of galactose into the yeast cell so that, of course, it can be metabolized. We come to the next part of this question. Scientists produce genetically engineered yeast cells. Underline. The gene coding for MSN2 transcription factor proteins and the marker genes coding for the green fluorescent protein GFP are transcribed together to produce a single mRNA molecule. The resulting MSN2 transcription factor proteins contain a GFP region as part of their structure and are called tagged MSN2 molecules. These tagged MSN2 molecules show up as green fluorescent spots when viewed using a microscope with a very high resolution. An investigation was carried out to compare the distribution of tagged MSN2 molecules in yeast cells when glucose is absent and when glucose is present. Read the question at least twice to explain this to yourself. The results are shown in table 4.1. Glucose availability, glucose absent, glucose present, mean number of tagged MSN molecules. When the glucose is absent, the mean number of tagged MSN is 3263 in the cytoplasm, 2012 in the nucleus, and a total of 5275. Now, when the glucose is present, then you have 1755 mean number of tagged MSN2 molecules and 632, and this is 2387. So look at the difference. Glucose absent 5275, glucose present 2387. In the nucleus we have 2012, then we have 632 and 3263 and 1755. So just look at the trend. Is it more, is it less? And then start to figure out the question. Now calculate the percentage of MSN2 molecules in the cell that are located inside the nucleus when glucose is absent. So when glucose is absent, it was 2012. In some show your working and write. So we have 52752012. So this is the simple working that we had to do over 5275, and this of course comes to 38%. Part two of the question was, when glucose is present, 26% of FSN2 molecules in the cells are located inside the nucleus. Now you know inside the nucleus you have DNA. So it should come to your mind immediately. Suggest why this figure is different from your answer to one. MSN2 in the nucleus higher when glucose is absent. MSN2's transcription factors bind to DNA in the nucleus. So nucleus has to be something. So transcription factors bind to DNA in the nucleus. Or kya? What else can happen? So it makes sense if you understood that. And when were they more? When they were when the glucose is absent. So this was again, you got this answer from the question. It wasn't something which you had to know beforehand. We come to the last part of this question, which is the C part. Another method of tagging molecules with a fluorescent color is to use monoclonal antibodies that have GFP attached. These monoclonal antibodies bind specifically to the molecule of interest. This is called immunofluorescence tagging. Suggest reasons why immunofluorescence tagging is not a suitable choice for this investigation. Well, naturally, because antibodies are too big to enter the cell. Making any monoclonal antibodies is very expensive. It harms the mice, and it's not a very monoclonal antibodies. You have to inject the B cells into the mice, and then you have to let them develop the plasma B cells, and then you have to kill the mice, and then of course you take out the plasma B cells, and then you fuse them with the cancer cells, and you become hybridoma. So all that, of course, that is all what we study in the AS level. But then of course you have to come up with a general thing. Why you see, whenever they say is not suitable, then you have to find out the faults in that process. So harms the mice, and immunofluorescence is only used in dead cells. So any three out of these, and well, you could have got away with even 1 or 2 marks if you could just come up with something very sensible at that point in time when you're taking the exam. Question 7. Structures, process, and compounds involved in photosynthesis. C4 plants includes carbon dioxide, all these names given to you. Now, complete table 7.1 by matching each description with one letter chosen from A to N to show the correct structure, process, or compound. You may use each number once. So, enzymes with a high optimum temperature naturally had to be PEP carboxylase, which is D. Enzyme in bundle sheath cell had to be E, which is of course the Rubisco. Then process that slows down the rate of photosynthesis was J, photorespiration, naturally because Calvin cycle stops and the Rubisco starts reacting with the oxygen. Compounds that release carbon dioxide into bundle sheath cell was F, which is malate, and then cells that stop oxygen reaching the bundle sheath cell was H, and H was mesophyll cells. So this is what we had to understand. That was a little, as far as I'm concerned, it was a little easy, but maybe you, I mean, I don't know which part of the syllabus you're struggling with. Some plants, such as rice, are adapted to grow when partially submerged in water. Describe and explain the adaptations that allow rice plants to survive in waterlogged fields. Now, again, that's a very direct question. And it says aerenchyma, so oxygen can reach the roots, grow taller to keep the leaves and flowers above water level, high tolerance to ethanol, so roots can respire anaerobically, and they produce more of the enzyme ethanol dehydrogenase, which is going to break down the ethanol which is being produced from anaerobic respiration, because ethanol is toxic. So it will not really form, it will not produce the toxicity and will not damage the plant. So these were the different points, and you had to give me any three of these. So this would get you. So this finishes this video, and thank you very much.